Welcome, everyone. Today I have Professor Robert Harris with me, who is a neuroimmunobiologist. He's a professor of neuroimmunotherapy at Karolinska Institute. He did his PhD at University College London in invertebrate immunology, so basic biology research. Then he moved for a postdoc also in London School of Hygiene, working more with mammals, with mouse macrophages, and their interactions with particular parasites. And then he moved to Karolinska, where he progressed today to do two very important functions. One as a professor of, as I said, neuroimmunotherapy and trying to come up with new ways of approaching therapies in neurodegenerative diseases. And the other role, which I think is also very interesting to talk about, is that he serves as a dean of doctoral education at Karolinska Institute and oversees and makes some important decisions on education of a lot of PhDs. From what I gather, it's more than 2,000 PhD students, and also offers supervising for supervisors, which is something that I'm not so familiar with, but I believe academia is quite in desperate needs of. Robert, or Bob, as you allowed me to call you, thank you so much for coming. It's great to have you here. And how are you doing today?
Very good. Thanks very much for the invitation. It's a pleasure to be speaking with you. It's a sunny day here in Stockholm, so everything's good with me.
You have a very interesting background. Could you tell me what does it represent for you?
Yeah, it represents a 30-year journey of exploration and reinventing myself several times, I'd say, as a researcher. So as you mentioned, I started off with a PhD project, which was a pretty impossible task to purify an agglutinin from the blood of African snails. The snails were so small, I used to get 10 microliters of blood per snail. So I needed to have a lot of snails in order to get enough material to be able to eventually, it took me 2.5 years to purify the protein, but I eventually did it. And then moved into other parasites at the London School of Hygiene and Tropical Medicine. So I first worked with schistosomes, then worked with leishmania, and then I moved to Sweden 30 years ago and started working with trypanosomes. Joined a lab that was basically a multiple sclerosis lab, but they had this pet project because African trypanosomes cause African sleeping sickness, so a neurological disorder. So I was sort of the ugly duckling in the lab, the only one doing this parasitology. But after a few years, we gave up when all the parasitology funding went to AIDS, HIV research, or malaria. And then I switched into autoimmunity, the same as the rest of the lab was doing. They do a lot in human samples as well as rat. I started working with mouse models and setting them up, and then went through multiple sclerosis, collagen-induced rheumatoid arthritis, type 1 diabetes in mice, so a lot of autoimmunity. Then moved on and started working a bit with tumor biology, started to work with brain tumors, melanomas, and then sort of went back to neurodiseases. Well, I never really left it. I just started to sort of expand my horizons. So now focus on neurodegenerative diseases, so Alzheimer's, ALS. ALS actually is my major focus, I'd say, but still chronic MS and gliomas we do as well. So anything incurable in the brain, that's what I like to do. You know, the last vestiges of what we need to do, where there's no hope out there. That's what I like to do in my research. So it's been a really, really fun time to learn all these different things. And even though if you look at my publication record, it looks a bit schizophrenic because it's like I do structural biology, I've done biochemistry, I've done all the things I mentioned. It's a bit like, what the hell do you do? But I do all these things. And the fun thing is that in each subject area, you learn something else that you can actually take in to the next one. So I love this idea of sort of comparing and contrasting and applying principles from one subject area to the other.
You mentioned these diseases are incurable. Are they incurable, or are we
currently
incurable? What are some promising directions that you feel are happening in this field of neurodegenerative diseases?
I'll ask the question by answering another question first, which is what's happening in medical science. And if you think about what's happened in the last 20 years, there have been fantastic advances. So with autoimmune diseases, we have very good medicines now. If you take them early, you can basically have tolerable levels or slowing down the causes of rheumatoid arthritis, multiple sclerosis, type 1 diabetes. That's fine. Transplantation biology, you can transplant multiple organs. You can even do face transplants now. So transplantation surgery is just amazing. If you think of tumor biology, we have cures, not just treatments, cures for forms of cancer which were previously uncurable. So these leukemias in children, ALL, for example, they used to be you're dead 10 years old. Now kids with CAR T-cell therapies are completely leading healthy lives. So this has all been fantastic. But then when you go to neurodegenerative diseases, Parkinson's, Huntington's, Alzheimer's, ALS, chronic multiple sclerosis, brain tumors, there is nothing. There is nothing out there. So this is a really, really unmet need, and it's sort of like the last frontier. And of course, it's the last thing that drug companies are interested in, because it's very difficult to go into the brain without causing any type of damage. So it's the last place, but it's the last market for research as well.
Let's go back to your beginning of the background, your PhD, which was, as you said, in these small snails that would give you very little blood, and you were trying to isolate agglutinins. What exactly were you doing and trying to achieve, and what did you learn from that PhD?
I learned many things from that. I got the first thing is I learned that the project that was designed for me was undoable. It took me about 3 months to understand that. I was supposed to use a chromatography system which would actually kill all the activity of the proteins. But fortunately, another PI, a young PI, had just been given an FPLC system that you could purify proteins using columns that you could use buffers that actually restored the activity. So that gave me a chance. And then it took me 2 years to work out how to do it, which is basically the following. So this agglutinin was one of the proteins that the snail just has a hemocoel that the heart pumps blood to the whole of the body. There are no vessels, so you can easily get the blood from them. But the hemoglobin, you either have hemoglobin or hemocyanin, depending on whether the blood is red or yellow. It varies from snails, species to species. And the hemoglobin is like 90% of the protein. So the key was to actually pull out that, and I used the lectin bead in the end with the right carbohydrate specificity to do that. And that then gave me enough proteins left in order to be able to split them apart. So that taught me an amazing thing about, one, perseverance. It took me basically 2.5 years to get to the first part of my project. And I learned lots of different techniques during that time. When I wrote my thesis, there was a huge monstrosity written on a typewriter because I'm so old, but it was hundreds of pages, and 100 pages is just all the methods that I tried. I didn't get all of them to work, but that was the only way I could try and substantiate that I've been doing something. But that gave me a fantastic portfolio of technical abilities, and I think that's actually what's made me attractive then as a postdoc, because I actually know a little bit about quite a lot, and then I'm expert in a few things. So that was a very good thing. The other thing I learned from that period was that I had 2 supervisors. There are one at the University College London, one at the Natural History Museum. He was the guy that used to go out to Africa and collect the snails. And 2 very different supervisors that taught me 2 very different things. My first supervisor, he actually announced after about 6 months, I'm going to be working from home because I'm writing a textbook. And then I didn't see him for 2 years. And then he came back and said, how's it going? And, not very well, because I hadn't been getting very good supervision. But the other one was really great. He took me all around the world. He was very high up in the organizations, wearing a suit all the time. I'm standing next to him in a white T-shirt and a pair of jeans, and he treated me like an equal and introduced me to people and really showed me. So there was this, I think that's where I probably got this interest in supervision, because I had 2 very different experiences of supervision. One that didn't really do too much for me, and one that, even though intellectually he couldn't do too much for me, he actually treated me like a very important person. And that's actually maybe that's been a sort of most decisive influence from that period, I guess.
When you approach supervising today of students, how do you bridge these 2 worlds of trying to sometimes obviously you have to give criticism, you have to tell the flaws, which is a very delicate thing to do to someone who is a scientist, who has an ego, and they're struggling with themselves, but also then having them be inspired and stimulated and pushed. So to be motivated by criticism rather than crushed by it, how do you do it? How do you approach that?
Yeah, actually, this is one of the most important training lectures I do, and I do it for both students and for supervisors and all sorts of leadership groups. But it's something I also try to implement in my own supervision students, and that's the following: that you need to develop trust, because if you develop trust and respect, then you need that forms the foundation of this basis of the giving and receiving of professional feedback. And the giving and receiving of professional feedback is so critical. Yeah, we have egos, and we're very, on paper, we're the most intelligent people in the population. It doesn't mean we're necessarily smart. And we're emotional and drama queens and kings all over the place as well. But it's your duty to both give and receive professional feedback in a respectful way. And this is the thing I really, really try and promote. So if you get trust from your students, and you need to develop that early, and you do that by being self-critical, because if you're saying, look, I can actually, and you invite feedback from them, and if they're giving you feedback, both positive and negative, then it's easier to give them feedback, both positive and negative. And that's usually the way they do. So the trust between a supervisor and a student, or a supervisor, postdoc, or me and the director of KI, is all about having professional respect and trust. And once you got that, then you're usually in quite a good situation.
I know I'm jumping back and forth between research and supervising, but I do want to go back to those snails. You were saying this professor was quite motivated to collect these snails, go to Africa even, to collect them. Why were they so special to investigate?
Well, they're vectors of this disease, schistosomiasis, bilharzia, as it used to be called, and there's a species specificity. So there are the genus of snails was called Belinus, and there are over 30 species, and not every species of snail would allow every species of parasite to actually, it wouldn't be a vector. So if you got the right parasite in the right snail, then they would develop, and then the parasites would come out of the snail. If you got the wrong parasite in the wrong snail, then the snail immune system would kill the parasite, and there wouldn't then be any production of new parasites. So that was the idea, was to try and understand that in order to be able to explain that specificity, not with any way of doing anything about it, but just because it was interesting. And what I found out was that but in the end of this 2.5 years work, was that every species of this genus had the same protein, the same basic agglutinin, but the glycosylation patterns varied, and that's what gave the specificity. So it was a bit cool. And it's just the cool thing is that these invertebrate biology, immunology is very simple. They have very simple cells. They have very few molecules, but they're actually really, really efficient, unless you get sort of too high bacterial loads, and that of course will kill the organisms. But they're really, really efficient, and this is like the primordial functions that we still have in the mammalian body. With C-reactive proteins and things. The pentraxins are very similar. So there's a lot of knowledge to be gained by looking lower down the evolutionary tree. And in actual fact, drug companies are mining, especially what comes out of the sea. There are fantastic molecules which have been isolated from all sorts of seaweeds and invertebrate animals there. That actually are really, really useful.
So you were mentioning, and I got stuck on that, which is this glycosylation pattern in this matching of snail and parasite conditions. Is it about the pattern, and is it a simple pattern?
Now we're talking. I graduated with my PhD in 1991. So the things that you can do with glycobiology now, and this is actually the fun thing of even looking at mammalian proteins. It's not the protein part which is interesting, but it's the scaffold of the carbohydrates which are on the surface, and that's what makes receptor ligand interaction so important in the mammalian cells as well. So the possibilities of investigating these things now are immeasurable. But at the time, I could just strip off all carbohydrates and still show that the rest of the protein was still the same. That was the level of my examination. The things that the kids can do nowadays, the new PhD students, is just stellar compared to the things that I had the possibility to do. So it's really, really cool to be a researcher, a young researcher at the moment when it comes to that technically. I'm kind of jealous, each time I take in a student, I think, oh, the things that you can do is like 100 times better than the things I could do. But that's just one side of it.
Yeah, but also the requirements have kind of gone up. So there used to be a possibility to just report one experiment without any statistics. So
I published my first paper in 1992, and in those days you had 4 figures, which were single figures, and 3 of them were important, but you used to have a 4th one because everyone, 4 was the norm. And we published in Nature Immunology a couple of years ago, and with all the supplementary figures, then when it came to data figures, we had 115, included in that one paper. So it's like the expectations, the requirements now, especially if you want top quality publications, is kind of crazy.
Which is why I very often enjoy reading older papers, because it seems to be centered around 1 or 2 main points. Like, we got this one big discovery and it's a simple piece of data. Look, I don't know, this glycosylation pattern is contributing a lot to this one particular process. This is how we did it. And it's very simple and straightforward and enjoyable to read. Whereas now I feel when I read a paper, I feel like I need a PhD for that paper to just read it, because there's always like 2 omics techniques that are done and very complex analysis and like 2 or 3 different models sometimes, and conclusions are very long and difficult to interpret for me. How do you approach just catching up with all of the technologies that are emerging and progressing so fast?
Yeah, just before I answer that, I just go back. I don't know if you know how Watson and Crick actually published the DNA helix findings. Do you know that?
Which part are you referring to?
They had 4 publications in Nature, back to back, or not back to back, but within a short period of time. The first one was just the hypothesis. The second one was the method. The third one was actually the structure of the DNA helix. And the 4th was then what the interpretation was. They got 4 Nature papers, which were separate letters to Nature for that. That wouldn't be, nowadays you give the 2 reviewer, they put everything in one and they still ask you for 3 mouse models to show it's worth that. So, and now I've forgotten the question. What was the question?
Just how do you approach catching up with all these technologies?
Yeah, I mean, you need to be an active researcher to do that. I haven't been working at in the lab myself for 15 years or so. So I get in touch with the technologies through my staff, through my students and postdocs, and I do my best to keep up to date on them. But to tell you the truth, when it comes to the omics things they're doing, I can appreciate the final figures, but I have no idea how they got there. The imaging techniques which are used nowadays, I did a lot of microscopy when I was postdoc, but it's like, the ways that you can do the analysis now are beyond my comprehension. So I do my best to I don't actively read too much. I only have 20% of my time actually as a researcher, 80% I have doing everything else. But I review a lot of papers, and in fact, this is my way of keeping up. So reviewing papers for journals or reviewing grants for organizations is a great way to then get a good update. So I do a lot of reviewing purely for that reason. And then, of course, if my students and postdocs sends me papers to read and they say, you have to read this, then I'll do that as well. But I think it's very important. It's actually something I'll talk about when we come to leadership as well, that the older you become, the more distant you come to the ground floor, but you need to be in touch of what's happening to understand. And while you don't need to understand all the techniques, if we're talking about research in complete detail, you need to understand the possibilities, what that they present and how that might be useful for your research group. So you need to do that, and you can partly get that by attending conferences as well, where there are new innovations there. But it needs to be an active process if you really want to be a finger on the pulse type of researcher. and make good collaborations, because the sort of the whole way we do science now has changed. In the old days, you used to send someone to a lab to learn a technique, and then they come back and set it up. And now you just send the samples, and they do their bit, then you do your bit, because you haven't got time to waste doing that. So I think science has become a lot more collaborative, and that's why it becomes so multidisciplinary, which is kind of cool. We used to be, very much like silos, and very much insular in our way of thinking. So it's another way of doing it. So you need to, part of the knowledge is actually knowing where the techniques are for the people you can collaborate with, as opposed to actually being completely knowledgeable about everything yourself.
You said your research load is now about 20%, and you mostly do a lot of other things. But I guess you still have an active lab,
Yeah. No, it's still a very active lab, and it's actually doing better. We've been publishing extremely well in the last few years. I got the top grant in neuroscience from the Swedish Medical Research Council 5 years ago. So we're actually doing very well, which maybe says something about the level of the research when I'm not doing it with my hands. But no, I facilitate the research. I have the ideas, I write the grants, I provide. And I have two team leaders who are running the day-to-day things. One doing the wet lab, one doing the dry lab, and they're the bosses, and I have a lot of interaction with them. So yeah, that's the way it works. So it's very productive.
Living the dream, I would say. I think when I think about science and what aspect do I enjoy about it, it's actually this part, which is like bouncing off ideas and trying to understand like which idea is good and eliminate the bad ones, and then exploring them. But then when it actually comes to sitting at the microscope and pipetting, that's the part and your neck going really bad for a period of time. That's the part I don't enjoy. So how do you compare these roles as you progressed and got
Yeah,
to where you are?
you need different skill sets and when you're junior, you always want to get up to the next level. So when you're a PhD student, you want to be a postdoc. When you're a postdoc, you want to be a group leader. But with that becomes a lot, your job description changes when you get the professional responsibility. So lots of people get a bit shocked. Being a PI, a principal investigator, is a different thing from being a group leader, because as a group leader, you have a certain amount of administration, you have responsibility for workplace, the working environment, and how people are feeling and everything, which you don't have when you're a PI. It's just to do your research. So I think you learn different things at different levels. And that's the fun thing about the academic journey. Although on the other hand, I've not received too much formal training about how to actually navigate that. And I think many of us just do it by looking at people who are older than us and just seeing what works and hoping it's going to work for you. But the world is changing. The sort of research is quite cutthroat now and extreme. There's so many people doing research now that weren't doing it before. So the competition is crazy. So one needs to use those resources. So I've gone from here. I still have the ideas. I still have things I want to do, but I leave the details about how they're done to my staff. And it's really whatever they think is good. Sometimes they don't like my ideas. I just had a brainstorm with another group. They invited us in, another microglia lab, and usually when I turn up at my place of where my research lab is, even though it's 20% on paper, it's usually just half a day, which is like 10%. And now when I turn up, people often call me a stranger, like, "Oh, we have a nice visitor today," because I'm not considered to be like part of the team anymore. But anyway, and you're considered, you're old, you don't know anything. But I had a brainstorm with another group the other day, and I came up with a completely different take on their results, something that they hadn't even thought about. That they really think this is actually maybe the answer, and it was so you can still be creative, and I think that's my. I wasn't actually, if I'm honest, I wasn't that good at research. I could purify proteins. That was my expert skill, my superpower. But other than that, I wasn't like so fantastic. I didn't do my early days. I didn't do anything outstanding. But I'm an ideas person, so you need to have a creator, and that's sort of more of the job. So with age, you go through these different phases where you wanted to get to the next level, and then when you get to the next level, you think, oh, it's not as fun actually, but then you have a new challenge. So as you move through the academic ladder, you're learning so many different things, and now it's more about being a leader for the people that are running the research as opposed to actually calling the shots themselves. And that suits me fine, because then I can just be creative and see if I can convince them to do my crazy ideas.
You know how sometimes they say that the outsider is the person who can ask the best questions because they're just coming with a new, fresh perspective? To me, it feels like you're the ultimate, the best possible outsider because, first of all, you changed fields multiple times. You did so many different techniques. You worked with invertebrates and then mice and like different perspectives of immunology. So you are an expert in so many different things and you have compiled so much knowledge, but then now you're so, a little bit in the lab and you come as a very, very, very educated outsider. So I think that's like the best use of time. Like with that
Yeah.
10%, you come and bring a fresh perspective that is super educated and
Yeah.
backed by a lot of experience.
I think it's something that we do wrong in academia as well. We have so many old senior, let's not say old, but senior people that don't bring on the next generation. I often ask people, "What's your legacy?" And the legacy is not your publications, because they're like rockets that just go up and they're down, and you've forgotten them like the next year. But people, it's all about producing people, producing scientists, and that legacy can continue for a long time if you train them well, if you treat them well, if you infuse them to be scientists or whatever they end up doing. And I think that then it becomes less about the science and more about facilitating the science and making it an enjoyable experience. It's all about leadership, and if you're a good leader, then you'll actually get good work done out of your people. It's one of the most simple aspects of leadership, and I think so many people miss it. That happy people will work, and unhappy people will be unhappy, so they won't work because they're thinking about that they're unhappy. So if you make people happy, provide a good environment where they're seen, heard, respected, you spend time with them, then they'll do the world for you. And this is really what I try and bring into both the research and the leadership trainings that I do.
What are some things in your lab that you're excited by recently? What are some ideas that you and your lab are pushing to try to understand neurodegenerative diseases?
Yeah, so I'm always excited by the work we're doing, not just because we're doing it, but I think it's really cool. I tend to do sort of off the wall things. So there are two things which are really exciting at the moment, and they're both linked to the immunotherapy, because I'm not just interested in understanding how a disease works, a neurodegenerative works. It's like, I mean, that's useful, but I want to know how it works in order to be able to do something with it. If I can make the life of one patient better before I retire, then it's all been worth it. So it's very much designed. What we're trying to do is to work out immunotherapies to actually change the course of disease, and we do that by studying our animal models. So we have two approaches at the moment, which are really cool. The first is a molecular approach, where I have a great collaboration with the guy here at KI, Björn Höberg, that makes DNA origami. So if you think of folding paper as you did as a kid to make shapes, you can do that with oligonucleotides. And then you use these as vehicles to deliver drugs, silencing RNAs, whatever you want, whatever's compatible with the chemistry. And Björn's been doing these for a while, and then we said, well, I'd like to make them myeloid cell specific, so we can bind specifically the microglia in the brain. So we coated them with beta-glucan, which is a receptor from yeast, which myeloid cells have receptors for, like lectin-1. And so we actually made this targeted delivery molecule where we put a drug in. The first instance, it was a top 1 isomerase, so it shut down inflammatory responses, and we could show a beautiful reduction in multiple sclerosis in a mouse, at least. And I think that Björn has gone on and engineered these things in a cancer setting where they become activated at low pH. So you have really targeted delivery now, and you can target then different tissues in the body, and then you can target different cells if you start to coat them like we did. So that's really, really cool. And even though, the legislation for getting drugs approved, the first silencing RNA was only approved last year, I think, even though we've been working for them for like 15 years. So it's a bit slow in the legislation, but I think this is a really interesting prospect. And I think that this type of targeted vectors, people are using vesicles, which are sort of coating to make them targeted. There are all sorts of approaches. I think this targeted approach, as opposed to just chucking something in the blood and hope for the best that it ends up in the right cells, is one thing. So that I'm really fond of. But the thing I'm most fond of at the moment is a project we're doing with a colleague called Roberto Gramione. And he worked with a special type of stem cell called the amniotic epithelial cells. And these are cells which are in the amniotic sac. So they're producing lots of, when you have a developing baby and you purify them from the placenta post birth. And these cells then produce a lot of immunosuppressive cytokines, interleukin 10, TGF beta. And that's been a focus of what we think are molecules are important for suppressing pro-inflammatory neurodegenerative diseases. So these cells are so incredible. Human cells, we put them into mice, we inject them into the brain, they shut down multiple sclerosis in a mouse. And we just like to go straight into clinical trials in ALS patients because they have no treatment that's working for them. And I'm sure that these things will have a great effect. We've characterized the molecular mechanisms, what happens when they go into the brain, and they're just like completely shifting the immune system to be immunosuppressive there. So I think this has so much potential. The fun thing about these is that they've been used therapeutically since the early 1900s. So when people had bad burns on their skin, and someone came up with the idea that you need to sort of cover it, so they use these amniotic sacs, the whole membranes, and put them on, and even put small pieces into eye to cure eye disorders. They've been used for over 120 years, but now you don't need to use the whole dirty sac. Instead, you can just purify these cells, which you can freeze, and they don't form tumors. Like other types of stem cells, the mesenchymal stem cells of people, everyone else is working with, and these are sort of considered to be the poor relative. But I think this is going to revolutionize medicine. I think there will be a Nobel Prize there for somebody, and but more than that, I think that we just need to do this type of work, and that really excites me because this is actually this tangible. This will be something that goes into clinical translation, and that's what I've been sort of hoping for all my academic life. So I'm really excited about those two things at the moment.
It's fascinating what you're talking about. But actually, I think I'm quite opposite of you when it comes to like the approach, when it comes to just why you want to understand things. You said you don't care at all about just understanding this disease. You care about understanding it for the purpose of curing it and doing something about it. Whereas for me, it's like I have very basic interests of like how does a neuron fall apart when it's injured? What are the molecules and that are governing it? What are the pathways? And the motivation doesn't necessarily come from trying to make improvement in people's lives, but just curiosity with being fascinated with a particular biological problem.
Can I explain why though? Because I'm not a clinician. Even though we work in the environment, the Center for Molecular Medicine, where our work is half clinical and half preclinical, so we have this really nice environmental match. But I'm not a clinician, and I've always worked with my molecules, going from molecule to cell to animal model, and we do a lot of animal work. And I'm just like you. I am interested in the mechanisms, but the big thing that changed for me is when you meet patients. So I was invited, I get funding from a Norwegian ALS society, Alltid Lite Sterkere, and they gave me funding a couple of times, and then I asked them, well, is there something I can do for you? And they said, well, why don't you come up to Oslo? We have this like open day once a year where we explain to the society what ALS is, what treatments are, what we can do, so you could come up and talk about your research. So I flew up to Oslo. I took a taxi to this hotel where this thing was, and that was my first encounter with both ALS patients and their carers. And I came out of that session a completely changed man. I came back on the flight from Oslo to Stockholm, and I said, I need to work with ALS because these people are just amazing. And I think then when you actually get the sort of conceptual, if you get an ALS diagnosis, then you basically got 3 to 5 years on average to live. And it's a horrendous disease, but, and when you get into your later stages where you lose muscle function, you end up like not being able to breathe because your lungs start working. But before that, you lose the ability to swallow. So every 10 minutes, someone has to come and suck the saliva out of your windpipe, otherwise you'd drown in your own saliva and things. And so that this is like these most amazing carers caring for these patients 24/7. And then the patients, there was one guy there, his name was Paul, and he could only move his eyes. He was so advanced, and he had a computer that then he could write, and he wrote poetry. And then he was there on stage in his wheelchair, and his son was there reading his poetry. And his last book was called "The Joy of Life." You know, and this is a guy that's only been able to move his eyes, and he's still talking about the joy of life. And his eyes were just a light, and that had a great effect on me. And that's why I said earlier to you, I don't care how it works or what it does, as long as it has an effect, because just retuned my thought process. Obviously, if you understand the mechanisms, then we're going to be able to design better immunotherapies in order to have that effect. So the two things go together. But that's just why I commented the way I did.
Yeah. So you were talking about this very promising treatment you're working on in your lab, and you were saying that the phenotype in mouse was it for ALS or MS or both?
We've done the MS, and we have ALS on the go at the moment.
So you were saying that the phenotype completely, you're able to reverse the disease or shut down the disease. What do you mean by that in the mouse?
The mouse doesn't get any worse. It actually gets better. So we seem to actually do some type of regenerative processes as well. And that's one of the effects I think these cells have, that they kickstart the healing process. I think immunotherapy has been very, many in the last 10 years or longer, almost all the immunotherapies are based on antibodies. They're small molecules because they can be high throughput processed, and it's kind of easy to do. On the other hand, it's very dangerous to put antibodies into a body because they're very difficult to get out. There was this trial in the UK, anti-CD21, where they had these terrible adverse effects, and as a result of that, the British Medical Association said you should always be able to reverse the therapy that you do. Very difficult when you put antibodies into somebody. But otherwise, these other approaches, I think, could be a lot better.
What I'm hoping is that you guide me a little bit on the phenotypes that you can kind of try to heal, or these motor phenotypes.
Yeah. So what we do is what we've been able to show in our latest manuscript, which has been sent in for publication soon, actually, is that by putting these amniotic epithelial cells into the brain, what we do is we completely change the microenvironment. So the resident microglia become immunosuppressive. We actually induce Th2 T cells, which also have immunosuppressive properties. So we're completely changing the microenvironment, shifting it towards an immunosuppressive phenotype, and that will then reduce the pro-inflammatory mechanisms driving the disease. So the mice actually, they don't get completely cured, but they just don't develop as severe disease. And I think that's for all neurodegenerative diseases, we can't think of a cure happening like this, but the very first stage will just be slow it down. I don't know who you have as a favorite Nobel Prize winner, but mine is Julius von Jauregg. Do you know who he was?
No.
He got the Nobel Prize in 1908 for something called fever therapy. So he was head of the clinic in the neurological clinic in Vienna, and he was trying to deal with patients with neurosyphilis, which is caused by a parasitic spirochete. And there was no treatment, and you'd end up in the mental asylum, put away because you go crazy. But he was a good scientist, and he'd realized that he could see something with using these primitive microscopes. There was something moving in the blood of these patients, and if he heated it, then they seemed to not move as much. So he thought that heat is good. He had soldiers coming back from whatever war was ongoing in Africa at the time, coming back with malaria, obviously having fever. So he did the following. He took blood from a patient with malaria, a soldier with malaria, put it into a patient with neurosyphilis to induce malaria. He allowed them to have 3 rounds of fever, and then he could treat the patient with quinine. And what this did is completely stopped the development of the neurosyphilis. It didn't cure them, but it just stopped the progression. And it was called fever therapy, and he got the Nobel Prize for it. It was used up until the 1940s, until the antibiotics came, and now you can treat those things a different way. But I think that conceptually, this idea of just stopping or slowing the development is the first thing we're after. It's sort of healing, complete repair. Do we really think this is going to be possible, like in an Alzheimer's brain? I don't think so. I think we're going to have a window of opportunity early in disease, and we've certainly learned that from multiple sclerosis. Multiple sclerosis is very difficult to diagnose because you start off with a headache and sort of just a feeling of malaise and maybe a bit tired, and you can just think it's like a yuppie flu or something, but it's actually your first lesions on the go. So in Sweden, our clinicians have developed a routine now that there's a good drug, which shows as soon as you think you have MS, then you start the treatment. And this has completely slowed down the trajectory. It doesn't necessarily stop it for every patient, but early intervention just to slow things down. And I think if we can get these things just to slow first, that's step one. If we can get them to stop progressing, that's the second thing. And the third thing will be, can we fix can we actually repair? Can we promote remyelination? Can we get nerves to regrow? And then, I mean, that's the holy grail. But I think that if we can do step one first, that would be great. We need something to work for neurological diseases. And you mentioned earlier, like what's going on. That's cool. There were so many clinical trials being done in so many neurodegenerative diseases. You know, there've been 550 preparations tried for Alzheimer's. And every time they come, unfortunately, with so much hype that, you know, now we have the antibody that's going to work. There's one on the go at the moment. Doesn't seem to have really significant effect, maybe only in a small proportion of patients. So we need something. We need a success story in the neurodegenerative field. Otherwise, people are going to sort of lose interest. And maybe it will be with Huntington's, CRISPR-Cas. You've got one gene that's causing the disease, so you can cut that out, replace it. Maybe that will be the first one. But we need some type of success. And certainly with MS, then, you know, it's a treatable disease now if you get it early. But once it goes into its chronic stage, nothing there. So we're getting there. But I think we need to keep you need to keep realistic. We're not talking about cures here at first stage, but slowing down would be great.
A lot of these neurodegenerative diseases have the underlying immune component, and you said that you're talking about this window. If you can make, for example, microglia immunosuppressive rather than pro-inflammatory, you can slow down at least the disease for some time. Could you try to give me your best view on like the order of things as they happen in neurodegenerative diseases? Because obviously at some point there is a strong immune component that is pushing the disease. And if you can, as you say, if you can push it down, that slows down the disease, which suggests that majority of the driver of progression of the disease is immune. But what makes the immunity go up in that way in the first hand? Is there
That's a great question,
what are the
but
best guesses?
there's no answer to it. You know, we don't know the etiology of any of the diseases. We know that there's a genetic component. We know that there's an environmental component. And I think that the immune systems in different people have different thresholds of activation. So some people are like, you know, pro-inflammatory. These people tend to be more autoimmune. Other people tend to be a lot more anti-inflammatory, and maybe they're the ones that develop cancers more often. So, you know, there's this variation. We know that there are like over 100 genes which have been implicated in spontaneous ALS. So it's about the same number in MS. They all have small effects, but you put them together and you come over this threshold of activation. It's like game on disease. But we don't really know anything about why it starts. I think MS could well be sort of, I think with your brain is so important, your central nervous system is so important that's why you have immune cells, microglia, and so on to sort of be patrolling, to take away the garbage, to make sure anything bad doesn't happen there. But if you get bacteria, viruses in, maybe you get this focal response. And maybe if it's a little bit overzealous because you're trigger happy pro-inflammatory, then you get this first damage. I used to love watching Western films when I was a kid. Sat on the sofa, dressed up with my guns and you know, shooting the Indians on the scene. But there was always the thing I remember from Western films is there was always a fight in the saloon. And it was often like a big mirror behind the bar, and someone would always get thrown and it would be smashed up, and the sheriff would come and take everyone away, and you just look at this wrecked room. And I think this is potentially what happens if you get infections in the CNS. You get these focal areas of damage, and the damage then means that you get the chance of post-translational modification. You get alterations of proteins, and this is how you lose tolerance. So we've done a lot of work showing that this actually occurs in autoimmune diseases experimentally. So I think that that could be one reason. And once you've broken tolerance, then the immune system starts attacking itself, and then it becomes self-perpetuating. So you don't need the virus or the bacteria anymore. Otherwise, it's intrinsic the homeostasis is not working. There is no control, and so something happens, but we don't know those triggers. Our work in ALS at the moment is extremely interesting. I'm working together with a fantastic clinician, Per Roos, and he has this idea that metals are really important in causing ALS. And if you look at the epidemiological data, it's actually very convincing. There was an eruption in the volcanoes in Italy recently. So there's a hotspot of ALS around Mount Etna. Because what happens when a volcano erupts is all the metals from the earth come up in the air, they come down into the drinking water. If you look at car manufacturing places in Spain, they have these very big industrial areas where you do a lot of metalwork, hotspots of ALS. And many professions where you actually come into contact with metals, you have a higher risk for ALS. So this is an intriguing thing that maybe we need to understand how these metals then trigger the start of the disease. That will be one interesting factor, which will basically say, well, then we need to think about how we reduce our exposure maybe, but it's still not going to help you when the disease is going. So that was a very long answer to your question, where the real answer is we don't know. We just don't know why these things occur, and they occur for different reasons in different people, because not all patients are the same. This is one of the most difficult things with clinical science and clinical research, is that, with our mouse models, we know that this mouse will develop this type of disease and this mouse will develop another type of disease, genetically different. We can explain that. With patients, we can't. And so we need to translate what we know about the variation in animals into humans. So we need to work out which type of ALS patient is like a SOD1 mouse and which one is like a another mouse. So to try and make that connection.
So even if there is some kind of genetic component and predisposition for any particular individual to develop any of these diseases, so let's say I have a gene that will make me some number of times of higher likelihood to get Alzheimer's, for example. Obviously, these diseases a lot of the times come either with the motor decline, but also the cognitive decline. Right. Is there any the intervention when it comes to prevention, like these cognitive games or playing chess, reading, that actually work, and to what degree do they work?
Yeah, there is quite good empirical evidence now. That's why on your phone you get so many adverts and saying neuroscientists promote this. Yeah, anything you do to keep yourself working, your mind is going to help you. There was a very nice study. We collaborated with a university in Japan, University of Tokyo, and they had a nice study there where they're looking at people. In Japan, they have the oldest population in the world, so many people end up in old people's homes, being taken care of, and they often become very labile then because they're not stimulated. And so they do a very simple, everyday word association, visual association, just to sort of keep the cognitive processes going. And then they see patients have completely changed their behaviors, become cognitively more aware, become more socially interactive, and it's a really, really interesting thing. So I think that's one thing, to keep yourself active, and certainly it's very easy to do that now if you're playing games on your phone, very simply to do. But the most important thing is to understand, we know with aging, from the age of 50, you start to lose brain volume. So the decline in brain volume is exactly the same it is for an MS patient. It's just that it's like 20 years difference. So, there's nothing you can do to prevent the deterioration, but you can slow the process. The only factor I know which actually sort of protects you is intelligence. So the more educated you are, the idea is the more neuronal connections you've made. So when you start to lose them, you still got a lot more left. It's like having more money in the bank and you're making withdrawal every year. So the more you start with, the better. But the other thing is, we don't really understand about healthy aging. But what we do start to know, it's about healthy living. So the things you do during your early life is actually you're going to pay for later. And I think this is a critical thing. And the most underestimated effect there has to be sleep. I've been a bad sleeper for the last 35 years or so. Since I was a teenager, I've been on to bed at midnight and so on. And in the sort of recent years, when I was sort of struggling as a group leader and stressed about whether I'm getting the next grant and things, I was sort of averaging 4.5 hours of sleep. It's not very, very good. So last year, I actually listened to a wonderful podcast from Huberman Lab, all about sleep, and actually learned some tricks about how to do it. For example, I had no idea that a cup of coffee will have a 10-hour effect in your body. So I don't drink coffee after midday anymore. And I go to bed earlier and I take melatonin and I like cracked it. I'm sleeping like a baby now in comparison. And
It's very,
I just
it's quite,
wish I'd
yeah.
started doing it 20 years ago. So I think that, anyone listening to this, get your sleep in order, because your brain really needs this in order to reset and to there is a physiological mechanism. When you sleep, the three layers of your brain, they actually move apart. So the flow of cerebrospinal fluid is actually greater. So it's like a wash cycle. And this is why you need to actually sleep during the night. So for anyone who's a shift worker, doing the night shift, unfortunately, you don't get that effect when you sleep during the day, because then it's back like this, circadian rhythms. But I think that if we learn how to be healthier in our early life, we'll probably have a better older age life. And that's going to be the key.
But for some of these little practical changes that can have a big impact, you think, well, I think, or I thought, it's coffee. I've been drinking coffee since I was 8 years old. Well, that doesn't do anything to me. And then, at some point, I was struggling. I mean, I was getting the hours of sleep, but for some reason, it just wasn't as rejuvenating as I would hope it to be and recuperating. And I thought, oh, okay, let me try. I do most of the day I'm in the lab and I don't eat and I just drink a bunch of coffees. So every 2 hours, I just go to the cafeteria and have a coffee. So I thought to myself one day, revolutionarily, maybe after, let's say, noon, maybe I can have a decaf. Not a fan, but let's try. Shockingly, it had a big effect. So I'm trying to, even though I like the taste and smell of coffee, it makes me feel good and it keeps me going. I just switching to decaf after afternoon was a big deal for shifting sleep, and just these little practical things can have a big effect.
Yeah, and the older you get, the more sensitive you come to these things, because your body changes, your metabolism changes. So one needs to be mindful about this, but we need to have the right information about it. But there is some really good research that supports these things. So it's just to be aware and follow the good practices, I think.
I want to switch to the topic of leadership and supervising. This is something you actively do. You are responsible for, as a dean of doctoral school at Karolinska. You're, I guess, in charge of like how this process and how the experience of PhDs will look like. So could you just give me some basic principles of good education during PhD and good standards for supervision?
So I just mentioned in my role, and I have the ultimate responsibility for everything that's done within doctoral education. We have 2,300 PhD students, about 7,000 supervisors. We have 14 thematic doctoral education programs and 8 clinical research schools. So, everything, the way you structure it is completely critical. And during the years, I've had the privilege of developing doctoral education practices here at Karolinska. I made some important innovations in how we monitor progress of students and assume quality of the experience. And I also get to go out around the world preaching and teaching about this as well. So the interesting thing is to compare on a global scale. It varies so much. A PhD is not a PhD. It varies in form. It varies in length of time. It varies in how it's rigged in different countries. At least for us, then at the Karolinska, we are considered to be one of the top places in what we offer when it comes to an educational concept, in the way that we really put a lot of emphasis on supervisor training, both basic supervisor training and then advanced supervisor training as well. And we have very good quality systems for asking the questions that need to be asked. Should you be a supervisor? Are you actually taking care of people in a good way, and so on? So I think that the doctoral education experience should be an enjoyable one. It breaks my heart when I'm reading on LinkedIn so many people saying I had such a bad time. And of course, we started mandatory doctoral supervisor training here in 2004. So we've been doing it for 21 years. So we've had time to see the effects, the impacts of it. And while it doesn't mean that we all have perfect supervisors here at Karolinska, we have far fewer bad conflicts, and most people actually seem to be more and more pleased with the supervision they get and the whole doctoral education experience. So it's very much sort of balancing the education and the research. In the old days, we called it research education, and we call it doctoral education. But very much like the yin yang here, we got the research and the education. And for many people, it was considered to be completely separate things, and supervisors would say when we started to have taught courses as being mandatory part of the education, it's like, why? This is research. I can teach you everything you need to know about research. Why do you need training? But there are so many skill sets that doctoral students need to develop: communication, collaboration. You need to be understanding innovation, the ethics. There are so many things which are not going to be something when you're pipetting, fluids from patient samples or whatever. So I think that one needs to make sure one has a balanced educational package which actually comes into the research that you're doing, so that they're actually aligned, and that the expectations of what you do is not really about producing research. I usually ask people, how much of the research you did during your PhD ended up as figures in the publications that you might include in your thesis? We usually have a publication thesis here in Sweden. And no one ever gives an answer above 50% at the most. And in my field, it's probably more like 20 or so. So that means that 50 or 80% of your experiences are not shown anywhere, but they're experiences you learned. Research is all about searching again. That's why it's called research. It's searching again and again. So doing science when everything's working is not really, anyone can do that, but learning how things work. And for me, struggling for 2.5 years to get to first base on my own PhD project, that's where you learn to be a scientist. And I think that so one needs to think very carefully about balancing education and the research, but you also need to make sure that the research is compatible with a doctoral education. And there are certain research projects which are good for researchers, but not good for doctoral education. So you need breadth or depth. You either need breadth, a lot of different experiences, or depth. You need to stack up layers of complexity in whatever you're doing. If you're doing like epidemiology, for example, statistical analysis, there will be a depth. My type of work where we're doing different models, cells, biochemistry, immunology, imaging, breadth. And I think that's these are the sort of the messages which I usually tell people that would give you the possibility of allowing an individual to develop the skill sets that they're going to need either to be a good researcher in the future, continue in the academic world, or for alternative careers.
What are some things in your experience and in your communication with PhD students that they're most struggling with during their PhDs?
There are many different things. I think the whole scientific world is crazy with the expectations. We talked about like numbers of figures in publications and what you need to do in order to get like top publications. It's ridiculous. And really the people calling the shots are the publishing houses. They're the ones directing the research, because they're saying, you want to publish in Nature, you need to do this type of research. And the funding organizations that say, we want people who are going to publish in Nature, so we're going to fund this type of research. And the researchers are saying, well, I want to be successful, so I better do this type of research. What happened to free research? So I think that's a poor state of affairs. But students need to, you need to find a vocation. There's no like good, bad, or ugly research. It's whatever you want to do. And I think that a student now is got this tremendous expectations and weight of everything. It's become so serious. You need to do your courses. You need to be abreast of the literature. It's not a 9-to-5 job, So people are. We used to have students come in and they don't really know what they want to do, but I could do a PhD. I'll be safe for 4 years, and then I can work out what I want to do in my life. You're not going to get that type of person now. If you're not really interested in science, not really interested in taking that journey, then it's not for you. And it breaks my heart to hear people talking about resilience and resistance. You shouldn't have to sort of survive a PhD. It should be an enjoyable time, even if it's not productive. You can learn a lot, because it still means that you've amassed skill sets. And like producing these fancy publications in top journals is just the icing on the cake, But not everyone who wants to be a football player will end up playing for Manchester United, It's like a very few that do that.
Maybe not the best example these days.
Yeah, maybe not. I'm aware of that. But it's more that not everyone is going to make it there, but some people will. So it's like, why not? But I think you need to come in for the right reasons and with the right. But the expectations become very unfair. I'm pleased I did my PhD 40 years ago.
Do you think there's a way back to free research and an approach where somebody can simply pursue something for curiosity rather than being guided by this cycle of publisher, grant funders, and so on?
I think there can be, because there's still research people willing to fund that type of research. But mainstream, unfortunately, I'd say no. I think it's going to become narrower and narrower. Even within the medical field that I'm in, it's like we have that discussion here at Karolinska a lot. Should we focus on core areas where our strengths are, or should we allow everyone to do their own thing? And obviously, you have a competitive advantage if the metrics are what you produced and the impact and grant money in to actually be in the stronger areas. So I think that research is going to become a lot more narrow, but there'll always be people who think it's a cool idea. There are lots of private people funding research nowadays. I have the privilege of receiving private funding myself. And so if you find the right individual, whether it's like, studying that seahorses, which you can only find in three oceans in the world, or that, plant that only grows up in the Himalayas, then I think people will always manage to do it. And I think that's cool. But I think the majority will become a lot more, actually, the mainstream will be a lot more solid, I predict, which is a little bit shame.
But I feel that I am the type of, I don't know, is that just a personality trait that to get just interested by something very obscure? And I feel like I'm that kind of person, and that's definitely not necessarily mainstream. And you obviously see that, okay, like whoever's writing grants, it's a grant for this particular thing. So if you want to get money, you need to write about that thing and deploy particular approaches and so on. But at least during my PhD right now, what I appreciate about my current PI, Professor Bruno Lemaitre, is that he's that kind of guy. He can afford to pursue things that are simply interesting. So I'll give you an example. Right now, we're just working on a group of very short sequence proteins that are conserved only in the fly, not really anywhere else, but they seem to be doing a very immunologically interesting role in the fly by protecting them against their own very limited immune response, like not something complex as you can see in mammals, but they do seem to be protective against this very particular kind of autoimmunity. And we're just playing with it, using all the tools, and it's an interesting journey, even though, when the first paper on this topic was about to be published, he was, discussing with multiple editors, and they all told him, like, this has no conservation, we're not interested. But he still manages to pursue it, and simply because it's biologically interesting. And I hope there will be some place in academia for people who study a little bit off the shore things.
Okay, and most of what I've done in my research has been off. I had this coming from the field of parasitology and then going into the field of autoimmunity, and knowing about this fever therapy. Then I had this idea that why don't we use parasites to cure autoimmune disease? So I worked on that for several years. We published actually a couple of papers showing that if we injected schistosome eggs into rats with rheumatoid arthritis, we could shut down the disease really significantly. The same with the MS model in EAE. And we did that with a couple of different parasites. And when we published these in impact factor journals like 1.8, 2.1 or so. It was very difficult to get them published. People were saying they'd send them to a reviewer. One would be a parasitologist, the other would be an immunologist, and they'd understand half of the story, but not the other half. When I was at conferences, I'd always be like selected to present at the last day, end of the last session, when most people are gone home. And then I used to get a mixed response. Half the people came up to me and said, "You want to do what? You want to infect people with parasites?" And then the other people would say, "This is genius, go for it." Anyway, fast forward, these crazy ideas that I never got research funding for when we published in a very low quality. and someone else did, and now it's become a standard therapy for inflammatory bowel disease. You can actually buy parasite eggs over the net, and you just drink them with water. They hatch from the eggs. You get a pig parasite serous infection in your gut for about a month, and then your immune system will actually take care of it. And 50% of the kids are completely healthy afterwards. So not so crazy ideas. So I think it's really, and I remember coming back from a conference when I first heard about Siglex. So this is sialic acid bearing lectin receptors, and I, with the history in glycobiology, I thought these are so cool. And there was this Scottish guy talking about them. He was the expert, and I was like, oh, this is brilliant. So I wrote a grant to the Swedish Medical Research Council. Didn't get funded because no one knew what a Siglex was. So I think that the research field is like very much like they're not there to allow the really creative ideas. I got funding once from the Juvenile Diabetes Research Fund. That was cool because they have an innovation fund where you just need to have an idea. You don't need to have any data, and I put in an idea that I'd be transferring immunosuppressive macrophages in order to cure or to treat type 1 diabetes, which we did. Worked really efficiently in mice. And they actually, I remember they wrote back to me and said, we're going to give you the funding, but our scientific committee doesn't believe in your idea. But they still gave me the chance. And that was like really, really cool because I actually proved it worked. I never got like sequential more funding for them for some reason. But anyway, I think this sort of idea of cultivating, allowing a certain amount of innovation is what we need. And the boring thing would be if we do this more centralized, we'll only do everything the way everyone's doing, then we lose that creativeness. Now we have so much bioinformatics where people are just mining data. Like there is some creativity in that, but it's very much like we're in this technical bubble with all the omics. Hopefully we're soon out of it, where we're just thinking if you do, then maybe something fun will come out of it instead of having real hypothesis-driven research. So I hope we can balance those two sides in the future.
I hope so too, but I'm a bit discouraged by hearing a couple of times the saying, you get nature, you get a job. So no nature, no job. I feel like that's become a bit of a standard. I don't know. Do you think that's true?
No, I don't think it's true. I think that certainly it depends. I mean, if I'm hiring postdocs, I'm interested in the person and how they can communicate and what I consider their skill sets to be. Whether they're produced here, they're everywhere. We all know it's nice when you publish in top journals. We published several times in top journals the last few years, so it's great, and that means I have a better prospects of getting grant funding, possibly, or better chances of publishing again in a higher impact journal. But is it any better science than I was doing? Like my favorite paper is the one that we did when we first transferred macrophages, immunosuppressive macrophages, and showed that we could shut down EAE. It was published in a low-ranked journal, 2.4, and that's the best piece of work I think has ever come out of my lab. I mean, so I'm the one I'm most proud of. It's not the best piece of work, the one I'm most proud of. So hopefully we go. I don't think it's and also because to publish in top journals, we have like 30 authors. The contributions of so many people, actually, then you're not sure what this person's role has been. Have they been doing a significant amount of this, or just because it's published in a top journal, does it really mean that they're that good, or does it mean that the cumulative team is that good? So we need to readdress that. So for me, I'm not interested in the prestige. I'm interested in whether I can talk to this person, whether they can articulate their science, whether I think they have skill sets. And I don't care if they publish in Nature or Scandinavian Journal of Immunology.
Preach. Please preach that to all of your colleagues.
It's up to every individual, but I think you can get fooled by those things. What I'm interested in is, most interested in is hearing people talk about science, because then I really know whether they're inspired to do this for the right reasons. That's my major criteria when I'm hiring.
Do you still manage to read some papers in invertebrate immunology and learn something from them?
Not really. I don't get them in my feeds anymore. I mean, every now and then I see a name I recognized from the time, and but not as much as I'd like to. Of course, like to is up to me what I spend my time doing, but it's nothing I prioritize at the moment. But so I'm very much out of date, but it would actually be fun to revisit that. I was asked recently to give a talk about my academic journey, and then I had to go back to all these early publications and find the journals and things, and that sort of re-sparked a little bit of interest in the field. But no, I'm not following it. I'm sure there's lots of cool things happening there.
I'm thinking that every scientist, they have their own main topic of research. They have their own main ideas that they push for in the grants, and they try to get money for it. But every now and then they have an opinion that is a bit against everybody else's opinion. So I've seen this with all of my PhD colleagues, with postdocs, professors. Everybody has some opinion that they don't really struggle to get money for. But they kind of just have that opinion, like the actual cause of Alzheimer's is not this, it's this, or that phenomenon. I think everybody's wrong. Do you have a hot take in science like that you just
Always,
think?
always. I mean, let's say curing autoimmune diseases with parasite infections. Everyone's just said you're crazy. It works. Doing immunotherapy. Everyone's saying, why don't you want to deplete T cells or B cells or transfer regulatory cells, to transfer macrophages? They're going to work a lot better. It will never work, they say. We showed it worked. Time and time again, and now we have these amyloidic epithelial cells, and then people are saying, well, why aren't you using mesenchymal stem cells? Because the whole world is using MSCs. So why are you using AECs? Because they're better. We think they are. So time and time again, I face this, and I think it's up to you as a scientist to just stay the course as long as you can. You have the possibility. Carl Sagan had this fantastic quote: every kid is born a natural born scientist, and we beat it out of them. Few trickle through the system with a wonder and enthusiasm for science intact. And I think that's very much sort of the case, not only for kids going through school, but the whole of the university and especially the PhD experience. So we need to get back to actually enthusing people to think, and thinking outside of the box is where the innovations come from that have had the most impact in science over the years. So why wouldn't we encourage that? So I think that there are no bad ideas. It's just bad execution. And science is all about having a hypothesis. This will work this way, and you either prove it or you disprove it. And we need to get back to doing that. I'm not worrying about. Obviously, it's easy to say. You're not worrying about your next grant, and you have to, especially when you're younger. Being a younger PI nowadays is a lot tougher than when I was a PI. I thought it was tough anyway. And I think there we need to actually make sure that the leadership understands how things are changed. This is one of the paradoxical things about leadership, that you get to be a leader in an organization, at successively higher levels, the older you become. And then you're making decisions not about your own future. I'm 58 years old, so I've only got like 9 years left here. And I'm making decisions to affect the generations coming after me. And so if I'm not talking to them and asking them, how do you find it? And still thinking like, it's really easy to get good research funding because I've got it all my life. It's not. You need to be updated. And I think we need to make sure that we modernize that. So hopefully this idea of the prestige of doing science, I'm just looking forward to when we stop publishing in journals and we just have an open repository. It's a bioarchive and that's it. They're doing it in the field of physics, been doing it for a long time. So why would you need to have journals and all this like prestige? It's just like out with your data, start interacting with the community. And get used to doing science that way. Hopefully that's going to be the future. That might be one part of balancing the prospects.
I was thinking to make that the last question of the podcast, but I'm so pleased with this way of thinking that you've cultivated throughout your career, of not necessarily being contrarian, but coming up with ideas that don't necessarily go with the current mainstream flow. So maybe the final question for you will be, how did you learn to generate those kinds of ideas and have the courage to kind of be innovative despite lack of maybe funding or lack of support or lack of everybody understanding that idea has potential?
It's a really interesting question. I never got that before. I think some people are naturally creative. and other people aren't. They actually say if you look at the theory for the most efficient teams, then you should have 8 people. You should have one person who's the ultimate authority, one person who's the manager, one person who's the creator, and 5 people to do the work. Okay, but one creator. You need one person with the ideas. I think I just always been I was always a dreamer as a kid. I had a good fantasy. I used to love writing as a kid, and I think the fantasy comes out there. So I think you're either born with it or you're not. And then if you have the chance in your adult life to actually then use that in your work. So am I like super creative? Not necessarily. You know, sometimes I miss things as well. When we started doing EAE in mice, we had some knockout mice which were on the right genetic background, and I thought, oh, it doesn't matter that they're knocked out. We'll just see if the disease works. And the mice were dead after like 5 days. And instead of thinking like, why is that happening? Then I was just thinking, oh, I have to make sure I can buy the wild type mice because that, you know, that wasn't what was expected. If we'd have analyzed that, maybe I would have identified interleukin 17 because that was the reason that in this particular knockout, and that's the reason the mice were getting so sick. But I wasn't smart enough to make the connection. So I think there are different types of creativity. One is having an idea that's outside of the box, or it's really, for me, it's more like, say, with this background that I have with all sorts of different subject areas, I'm just able to use that and say, well, I'm used to working with parasites. Let's try and put them into autoimmunity, see what happens. And that leads me to immunosuppressive macrophages. So what more can we do with that? And it's more like connecting the dots as a type of creativity or innovation as necessarily coming up with something completely, you know, that goes against the grain. But I think a lot of scientists don't actually ask questions anymore. They just do things because they know they work. So a colleague of mine here has just been asking, we do a lot of microglia depletion, and there are now antibodies that go against the CSF1 receptor. You can very simply and very fast deplete microglia. So these have been used in clinical trials. But he suddenly asked the question, well, what happens when you actually do this? What happens to the cells? No one's actually described it in the literature, how they die. And it turns out they don't die. They actually become activated before they actually end up dying. So all sorts of interesting questions there, just under the nose of people. And I think creativity then is maybe just having the good sight to see what the question is, as opposed to saying, you know, I don't wake up in the morning and saying, now I need to come up with a revolutionary idea about research. No, that's not the way it works. But the mindset needs to be that you're open, not closed. And I think that's the key.
I really like that message. And I recently read this quote by, I think it was Watson, who said that a scientist has to be slightly underemployed to do great science, which is kind of reminiscent to me of what you're saying, is that scientists now maybe are too burdened with experimental work and things that they have to do to get the grant. And the experimental load is high, administration load is high. So everybody's kind of overburdened, and there's like very little mental space to kind of exit out of that and actually look at like, what's the interesting question here, and can I flip the question a little bit and ask a better question? So yeah, I'll try to cultivate that a little bit. Thank you so much.
I think we spend too little time discussing science nowadays. People are like, you know, they're mice in their cages, and as you say, well, you're juggling balls and you get more and more balls to juggle. But the most interesting time that I enjoy as a scientist is when I'm just talking about science with someone. We're just brainstorming. We're just having a chat, and just, you know, I could spend hours doing that because that's the most fun thing. But I think one should actually prioritize time to do that with your peers, you know, with somebody that's in your subject area, and that's really where you get these cross fertilizations. I mean, they do this in society. You know, they have like casual Fridays, and in certain companies, they've said, you know, you can go and talk to anyone, you know, any department, and through opening up these possibilities, they start to make products that they never would have envisioned before, because you get people with different mindsets coming together. We need to prioritize time to think. And you can only do that if you're healthy, if you slept well, you got the cognitive ability. So if you're doing too much, then you need to learn how to actually be realistic about your time, and time management is something which we all have as a challenge, but that's also something as a younger scientist to be mindful of, and the faster you learn that, the better, I think. I'm a very bad example of that.
I feel like we haven't exhausted a very high percentage of things I would like to talk to you about, but I think that's a good reason for us to talk again sometimes.
Yeah, I'd love to do that.
Thank you so much for coming and talking to me, Bob.
Thanks a lot for having me.